Hello and welcome to e-lecture series of PharmaIgnite, an initiative taken by LG Institute of Pharmacy in the uncertain period of COVID pandemic. I am ARTI PATEL, Assistant Professor at the Department of Pharmacognosy at LG Institute of Pharmacy. Welcome to Semester 3. This is my first lecture in SEM 3. We will discuss about secondary metabolites of plant which are used medicinally. We will discuss about their chemical nature, their classification, their definitions, and general chemical properties and general chemical reactions of certain secondary metabolites. So let us start. In plant, there are two types of metabolites, primary metabolites and secondary metabolites. Primary metabolites provides nutrition to the plant as well as the human being, whereas secondary metabolites provide defense from microorganisms to the plant. Various viruses, bacteria and protozoas may attack the plant, and these secondary metabolites help in defense from those microorganisms. They also provide defense from herbivorous animals like insects, worms and mammals. Secondary metabolites also help in attraction of pollinators like insects, birds and bats, which is beneficial for the plant. Secondary metabolites of plants are useful for mankind as they exert pharmacological effects, sometimes toxic effects. But we, the scientists, determine the particular dose for particular pharmacological action, and we use them as the medication, as the medicament for treatment of various diseases. So, if we see definition of secondary metabolites, the secondary metabolites are metabolic end products of primary metabolites of plants. They are generally occur in the plant in the form of either alkaloids, glycosides, flavonoids, tannins, resins or volatile oils. So these are the categories of secondary metabolites you have in the syllabus. So we will study these categories of secondary metabolites in detail. So let us start with the glycosides. Glycosides are organic substances. They are non-reducing natural compounds. Upon acid or enzymatic hydrolysis, they yield one or more sugar molecules along with non-sugar part. They are present in lot of plants and certain animals. The functions of glycosides in plants are they serve as the sugar reserve in the plant, for the plant. They protect plants from the toxic effect of a glycone portion, which is non-sugar portion. It increases the solubility of a glycone in the plant sap. It helps in transportation of a glycone to the site of action. Certain glycosides influence the rate of photosynthesis, and they participate in regulatory, sanitary and protective functions of the plant. So these are the general functions of glycosides in the plant. We will study in detail regarding all these functions. Let us see the properties of glycosides. If we see physical properties, physically they are crystalline or amorphous solid compounds. They are colorless, but there are some exceptions. Certain glycosides like flavonoidal glycosides are yellow in color and anthraquinone glycosides are orange or red in color. The solubility, if we see, in general, the glycoside molecule is soluble in water. Glycoside means sugar and non-sugar in combination. The whole molecule is soluble in water, alcohol and ethyl acetate. It is insoluble in organic solvents like benzene and chloroform. Glycone portion, the sugar portion of the molecule, is water soluble. It is insoluble in the organic solvents like chloroform and benzene. Aglycone portion is soluble in organic solvents like chloroform and benzene, and they are insoluble in water. Aglycone portion show optical activity. Generally, they are levorotatory form. Let us see the hydrolysis of glycosides. Glycosides can be hydrolyzed by various ways, either by the moisture present in the herb itself or by enzymes present in the herb or plant, or acids or alkalis from the externally. If we treat the glycosides with acid or alkali, they will get hydrolyzed. They may get hydrolyzed with the oxidative reagents also. The hydrolysis of glycosides by moisture, which is present in the herb itself or externally if it gets absorbed into, it is either desirable or sometimes non-desirable. Desirable means sometimes the hydrolysis of glycosides by moisture gives beneficial effect. It may increase the pharmacological action of that particular secondary metabolite. And undesirable means sometimes the present moisture degrades the chemical composition of the herb, and ultimately maybe the active phytoconstituents turn into inactive phytoconstituents, and hence here we are giving two categories, either desirable and sometimes non-desirable. We will study in detail regarding all these properties. Chemical properties of glycosides, if we see, they are hemiacetals produced by condensation of alcoholic or phenolic hydroxyl group of organic compounds with the hydroxyl group of sugar with a loss of one water molecule. Here you can see the hydroxyl group of the organic compound and hydroxyl group of sugar. They get attached with, they release one molecule of water, and through a common linkage they are attached, and this is the whole glycoside molecule. Here one side the sugar molecule is there, another side a glycone portion is there. A glycone means non-sugar and sugar portion are attached with each other through a linkage. The secondary hydroxyl group of sugar condenses within the sugar molecule to form an oxide ring which is in cyclic form, and hence glycosides are also known as sugar ethers. So this is the graphical representation of the glycosidic linkage of hemiacetals. Now let us see what is glycosidic linkage. It is the linkage between glycone and aglycone part, means sugar and non-sugar part. On the basis of this linkage, alpha and beta glycosides are assigned. Glycone are the sugar portion and aglycone are the non-sugar portion, which are also known as genins. Aglycone portion are having varying chemical nature. They are of either alcohol form, they are also present in the form of phenols, anthraquinones, isothiocyanates, cyanogenetic nitriles, steroids, terpenoids, etc. So aglycone portion is having various chemical derivatives, whereas sugars are also of different derivatives. So the combination of this non-sugar and sugar portion gives us glycoside molecule. The aglycone portion is responsible for the therapeutic action of the glycosides, and the sugar portion is helping in the solubilization and transportation of aglycone portion to the active pharmacological site. So wherever we need the action, the sugar portion will help the aglycone portion to transport to that particular site. Now, classification of glycosides is based on various features. Like glycosides are classified in various ways, like based on number of sugars attached with the aglycone portion. Second type of glycoside classification is based on nature of sugar which is attached with the aglycone portion. Third type of classification of glycoside is based on chemical nature of aglycone portion. Then fourth type of classification of glycoside is based on glycosidic linkage of the sugar and non-sugar portion. And the fifth one is based on the physical and pharmacological action of the glycosides. So these are the 5 different ways through which, based on those different categories, the glycosides are classified. So let us see first classification, which is based on number of sugars attached with the non-sugar portion. So if one sugar molecule is attached with the non-sugar portion, the glycoside is known as monosides. Then if the aglycone portion is attached with 2 molecules of sugar, then they are known as biocides. If 3 sugar molecules are attached, then they are known as triocides. Second type of classification, which is based on the nature of sugar. Say if glucose is attached with the aglycone portion, then those glycosides are known as glucosides. If rhamnose is attached, then rhamnosides. Rhamnoglucosides, if rhamnose and glucose, both the sugar types are attached with the aglycone portion, then rhamnoglucosides. Then galactosides, fructosides, ribosides, and so on. Then next type of classification of glycosides is chemical classification, which is based on the chemical nature of the aglycone. As we have seen, aglycone is having varying chemical nature. So depending upon the aglycone chemistry, the different glycosides are classified. This is the most acceptable classification of glycosides, because it is easy to distinguish because of varying chemical nature and specific chemical nature. They are easy to subclassify because of their specific chemical nature, and the subclassification favors another type of classification, which is chemotaxonomical classification. You will study the chemotaxonomical classification in detail in another chapter. These are the different chemical classes of the aglycones, like anthraquinone glycosides, steroidal glycosides, triterpenoidal derivatives, steroidal saponin derivatives, flavonoid glycosides, coumarin glycosides, lactone, cyanogenetic, isothiocyanate, aldehyde glycosides, phenol glycosides, alcohol glycosides, xanthone glycosides, lignan glycosides, and ester glycosides. So these are the different chemical classes of glycosides according to the chemical nature of aglycone portion. Now another type of classification of glycoside is based on glycosidic linkage. Say if the first class is O glycosides. So here an oxygen linkage is there between the sugar and non-sugar portion of the glycoside. Here you can see this is the glycone hydroxyl and this is aglycone hydroxyl. They are attached with the O atom in between. So this is O linkage in between glycone and aglycone portion. Glycone is sugar, aglycone is non-sugar. These glycosides occur very common in nature. cardiac glycosides, cyanogenetic glycosides and flavonoid glycosides are mostly possess O-glycosidic linkage. Second class under this category is C-glycosides. So here you can understand the linkage between the sugar and non-sugar part is carbon. So the same way here the carbon is the middle linkage between glycone and aglycone. They are resistant to normal hydrolysis, normal mineral acid hydrolysis or enzyme hydrolysis. So we need oxidative reagents to break this C linkage. You will study in detail about how we can break this C linkage. But remember that C linkage is cannot be broken by the normal mineral acids and enzymes. We have to use oxidative reagents like FeCl3 or KMnO4 to break the C linkage. Cascarosides and aloin are the examples of C-glycosides. Another class under this category is S-glycoside. As the name suggests, you can understand the sulfur linkage is there between sugar and non-sugar part of the aglycone. The thiol group, when a thiol group is present in aglycone, the S linkage is formed and they are known as S-glycosides. Green is a compound which is isothiocyanate glycosides, which is present in mustard seeds. Next category under this classification is N-glycoside. So here nitrogen is the linkage between sugar and amino group of the aglycone. So mostly enzymes and nucleosides are the example. They possess the N-glycosidic linkage between sugar and non-sugar portion. Now another category of classification which is based on physical action. So based upon the physical action, glycosides can be classified into, like if the glycosides are having soap-like action, then they are known as saponin glycosides. If they are extremely bitter, then they are known as bitter glycosides. If they produce HCN on the hydrolysis, then they are known as cyanogenetic glycosides. Another category of glycoside classification is based on pharmacological action of the aglycone portion or overall pharmacological action of the glycosides. So the categories are cardiotonic, purgative, expectorant, local irritant, adaptogen and anti-ulcer. So these are the different ways we can classify the glycosides, but among all the ways, two categories of classification are used mostly. One is based on chemical nature of aglycone portion and second is glycosidic linkage, based on glycosidic linkage. Now coming towards the identification test of glycosides. So one category, saponin glycosides. Saponin glycosides are identified by froth index or froth number, hemolytic index and fish index. So these are the three chemical tests through which you can identify the presence of saponin glycoside in your sample. Another category of glycosides, that is anthracene glycoside, they can be detected by Bontrager's test or modified Bontrager's test. Bontrager's test is used for O-glycosides and modified Bontrager's test is used for C-glycosides. So the difference between these two tests and the detail of the test we will study in our online lectures. Then third category, that is cardiac glycosides. Cardiac glycosides are identified by Keller-Killiani test, Legel test and Baljet test. Then cyanogenetic glycosides are identified by ferricyanide test, localization test, Grignard reaction test and cuprocyanate test. So So these are the various identification tests we can apply and identify different categories of glycosides. Now these are the references you can go through for further detail about this topic. Hope you have understood the outline of glycosides. We will study in detail about all the topics in our online lectures. Till that, subscribe our YouTube channel Pharma Ignite. Thank you.